So we're really delighted to have Dr. Jean-Paul Soucy here with us today from the MNI. We are going to get a PET talk today, which is a nice departure for us because we are at an MR site and we've been pretty MR focused. Dr. Soucy is the medical director of the PET unit at the MNI. He's got lots of experience training in nuclear medicine as well as in neuroscience. He has research projects in dementia as well as looking at the stuff he's going to talk about today, so some cholinergic imaging and some work in neurodegenerative diseases as well. So we'll get started and we'll have lots of time for questions after. So.
Thank you very much. Well, thank you for having me here today and for attending in impressive numbers, I must say, so that's great. So indeed, I'm going to talk about one part of the work I do in neurodegenerative diseases, and I'm going to talk about cholinergic systems, essentially. Most of it will be on Alzheimer's disease, but we'll also go a bit to things close to Parkinson's disease. And the plan I kind of expect to follow will be along those lines here, okay? Brief review of why it's of interest to look at cholinergic systems. Actually, it's not that brief, but I think it's worth it. We'll then see what has been done in terms of imaging the cholinergic systems with nuclear medicine, why we did select to go with PET and not SPECT, and why we did elect to go with, in particular, one molecule, FeOBV, that we'll be talking about. It's one that we've really characterized quite extensively over the past 10 years, I would say, something like that. And then we'll conclude with what we've done, in fact, in humans studying Alzheimer's and Lewy body diseases being taken in the most general sense of Lewy bodies being present in the brain, not necessarily dementia or Parkinson, as you will see. And then I'll just have a couple of slides about what we could do to try to work with you in PET looking at those systems. So, acetylcholine, why is it of interest? Well, I guess that many of you will know about what I'm going to talk about, but it's nothing new, actually. There were papers back in the 80s talking about the cholinergic hypotheses of everything Alzheimer, the symptoms, the memory deficits, and things like that. And indeed, there was lots of evidence that there were anomalies in the structure and function of the cholinergic system that had been demonstrated in patients with dementia, which are cognitive disorder, as we should say now. And there's a correlation, quite a strong correlation between losses of cholinergic systems and the clinical manifestations. And they can be reproduced, the symptoms can be reproduced by pharmacologically manipulating the cholinergic systems also. So that's added to the interest. Results were somewhat varied. It's you're not going to be able to read it. And basically, this slide is a review in that paper in 1982 of the evidence that had been collected mostly through histology about cholinacetyltransferase activity in patients and in models of Alzheimer's disease. So the first part is aged rodents, and as you can see, there were people who found that there was a decrease, others who did not find any decrease. Then we had elderly normal subjects, same thing. But when you go to Alzheimer's, everybody found a decrease in acetylcholinesterase activity. So that was pretty clear, no controversy there. Receptors, muscarinic receptors had been the target of multiple studies back then. Way less studies, same thing, aged rodents, elderly humans, and Alzheimer's patients. In Alzheimer's, there was, in general, no anomaly of muscarinic receptors. Only one study found something. Models did have some aging patients as compared to earlier, to non-aged patients, or subjects rather. Quite a, 2-1, that's pretty much controversial, as far as I would say. Still, there was accumulating evidence that indeed something of interest was happening. There were some studies that started looking at different correlations between pathology and the clinical presentation of subjects. So here there was an autopsy of 12 elderly women, and they looked basically at acetylcholinesterase, and again, in different regions. The frontal F2, the second one, if I remember well, very small, was the cingulate gyrus. The first one on the bottom is a T1. And the very last one was the lingual gyrus. So most of the areas that they studied had a decrease in those 12 women with Alzheimer's disease, and it correlated well with their blessed dementia score. So there was a good correlation, except for the lingular gyrus. Lingular, I don't know how you say it in English. In French, it's gyrus lingulaire. So anyway, that one under the temporal lobe. So that's the one I'm talking about. So there were more reviews, and they did look a bit later, 1999 now. And what they found was that indeed there was evidence of an important role for cholinergic systems from all the literature that had been undated since the first paper in 1982, with acetylcholine being shown to be an important modulator of transmission with excitatory amino acids, obviously. That was quite clear. And more papers kept coming out as the years went by. So another one, reviewing again the cholinergic hypothesis, and this one showing that both histopathological studies and pharmacological manipulations could be correlated to Alzheimer's pathology, in fact, in Alzheimer clinics. And the symptoms could be reproduced even in young patients using blockage or stimulation of the acetylcholine system. So, clearly something was happening. They also recognized that cholinergic systems could be involved in non-cognitive behavioral anomalies, including in the deposition of plaques as such. Another review some years later, so the data keeps, popping back. There's a series of papers coming out every 4, 5, 6 years about the subject. Again, reviewing what happened again with psychopharmacological manipulations, showing that you can reproduce symptoms in young patients that are similar to those that you observe in Alzheimer's disease, obviously in a very acute setting, but again, confirming that there's effect on those systems. So really the hypothesis that symptoms are linked to some aspect of cholinergic function seems to be getting stronger and stronger as papers accumulate. And things got very interesting lately. A few years ago, there was this really remarkable paper where they managed to show that connectivity between a fair number of patients, this time healthy subjects, more than 100 healthy subjects and 100 amnestic MCI cases, there was a very high correlation between the volume as measured with MR of the minor nucleus with the thickness of the temporal cortex. So having a loss of cholinergic cells in the basal forebrain was linked to atrophy of distant temporal areas, which obviously was of interest, and it also was shown for the amygdala and the hippocampus. So there really is a loss of structure associated with that, confirming, I think, quite clearly that innervation by the nucleus of minor is important for trophic support, at least, of those areas and function of those areas. More recent study of the same type, going even more in detail. This one has been performed by people with Montreal Neuro. They not only look at the basal forebrain, they look differentially at the nucleus of minor, and then they look at the medial septal and diagonal band of Broca, and they could show that atrophy in each of those two groups correlated with atrophy in specific areas of the cortex, which are known to be associated to those areas. So again, there is a loss of cortical tissue associated with a decrease in volume of those specific cholinergic structures. And in fact, we even have a pretty good way of explaining why cholinergic systems are important in Alzheimer's disease and why they're vulnerable. The thing started quite a while ago by looking first at the expression of receptors for a nerve growth factor in cholinergic neurons. And it was shown quite convincingly that indeed those cells do have nerve growth factor receptors on their surface. Both the mRNA and the protein were detected in those cholinergic neurons. On the minor, again, medial septum and the agonal band of Broca. So they do express those. And interestingly, which became clearer later why that would happen, it was shown that if you block the expression of P75NTR, one of the two great families, if you want, of neurotrophic receptors in the brain, you increase the volume and the number of cholinergic neurons in those structures. So there's this receptor, which is for a neurotrophic factor, seem to be able to, in fact, inhibit the growth of cholinergic neurons. And it has been shown now that there's this group of low-affinity neurotrophic receptors in the brain that do activate apoptosis when they are activated one way or another. So that was an interesting result, and you'll see why in a moment. This was actually confirmed again with a second study, so it's a solid result indeed. You get a decrease in number. The other one showed a decrease in size. This one shows an increase in number, in terms of cholinergic cells when you deprive those cells of their P75 receptor. And the other thing that was shown was that actually amyloid does bind to that P75 receptor, and it can activate it. And this probably explains why those cells are so sensitive to amyloid, because in fact, if you stimulate only the P75 receptor, that's exactly what you get. You get apoptosis. NGF stimulates both its tropomyosin kinase receptor and P75 at the same time. So if you excite only P75, as happens with amyloid, which does not bind to the TRK receptors, you basically shift your cells towards apoptosis. So that's basically what was suggested by this first study. And indeed, it was shown that you would get a decrease in cells numbers when you only had stimulation of P75. Same thing here. Again, you have confirmation that all trophins that activate P75 only without coactivation of TRK receptors will result in loss of cholinergic cells. And amyloid beta, again, does exactly that. So when you have the presence of an imbalance between TRK and P75, you end up having a decrease of the number of cholinergic cells due to stimulation of P75 only. And indeed, this is exactly what happens in Alzheimer's disease. They start underexpressing TRK receptors and express only the P75 one. Multiple effects of that have been confirmed. Again, here, this is exactly what it shows, the loss of TRK receptors as opposed to P75, which indeed remains constant in those cholinergic cells, and which leaves us with cholinergic neurons that only express the P75 and therefore go directly to apoptosis because the only stimulation they have is of P75, including through amyloid. In addition to that, cholinergic neurons have a strange behavior towards amyloid. Most neurons do not accumulate much amyloid intracellularly. There is beta amyloid peptides in most neurons in Alzheimer's disease, but cholinergic neurons, when they looked at them at autopsy, had a tendency to really accumulate amyloid beta intracellularly, and there's no exact knowledge about what this entails in terms of toxicity or anything, but it certainly underscores the fact that cholinergic cells have a different way of handling amyloid than other neurons, and that might contribute indeed in Alzheimer's disease to their demise. Not only that, when you have those kinds of abnormal stimulation, you have obviously formation of tau in a hyperphosphorylated form and then tangles. That's part of normal Alzheimer's disease. But cholinergic neurons, which can be identified as containing a polyphosphorylated tau molecule with this antibody here, PS422, have been shown to decrease in numbers, so there are less of those cells, except that at the same time, there's also a decrease in number of cholinergic cells overall. So, and that's happening before there's any NFTs. So those cells start being dysfunctional at a pre-NFT level, which is different from other neurons in general, which only will start dying off as NFTs start appearing in them. So cholinergic neurons are more sensitive to amyloid. They handle amyloid differently, and they seem to handle hyperphosphorylated tau differently from other neurons. So they're definitely, it's not at all surprising that they would be so sensitive to very early events in Alzheimer's disease. Okay, so I hope that by now I've kind of convinced you that it was of interest to look at those systems. I mean, they do show implication in Alzheimer's disease and in neurodegenerative conditions. And they have been indeed the target of numerous studies involving imaging. And this is a pretty old paper, 2008, that looked at imaging of those systems. The different types of imaging that have been performed have targeted either cholinergic receptors, nicotinic or muscarinic, both have been targeted. Synaptic vesicles through derivatives of visamicol, such as FeOBV, but initially nobody used that. Everybody was using a molecule called IBVM, labeled with I-123 using SPECT imaging. And then there's been labels for acetylcholinesterase that were quite popular at some time. So that's a good variety. The one problem that started happening very rapidly with acetylcholinesterase is that it was shown to not be specific to cholinergic neurons. It's also expressed by pyramidal cells in the cortex. So when you look at it, you're not sure that you're actually looking at cholinergic systems. So that's it. The changes could be due to something else than cholinergic demise as such. Another review, looking at pretty much everything that had been done until 2016, so that's more recent. Essentially, a few studies on using visamicol derivatives, therefore looking at the synaptic vesicular transporter for acetylcholine. Quite a big number of studies looking at acetylcholinesterase, despite the reservations that we can have about it, and a significant number of receptor studies looking both at nicotinic and muscarinic imaging. So, is this a good way of looking at it? Well, there have been a few papers, and this one is particularly striking, showing that it's not probably a good way of looking at patients by targeting muscarinic receptors. Again, the first old study that I showed you that looked at previous studies also had not found any evidence of muscarinic anomalies. This one, 2016, if I remember, that's it. No, sorry, older than that, 2001. They didn't find any changes in muscarinic systems in Alzheimer's disease. So not a very promising target. And again, this is the slide I had shown you earlier where they found no changes on multiple other studies. So it's not a good one to look at. Cholinergic changes, well, there have been quite a few studies, also small studies in general, as most of those studies are. They found indeed anomalies in the cortex during aging, not in Alzheimer's, but they found in Alzheimer's decreased binding in the hippocampus. However, that agent has been used in other studies, and results are quite mixed. There's really no consensus that you will find anomalies in nicotinic receptors, which is what the older studies had shown also. Looking at acetylcholinesterase, again, a paper, an abstract, and I've been waiting forever to see whether the paper would come out one day. It hasn't. So there might be something that you can't trust about this one, but it's just an abstract, where they looked at, no, sorry, not acetylcholinesterase, choline acetyltransferase. And what they did is that they measured levels of choline acetyltransferase in different brain areas in patients with Alzheimer's disease. And what they came up with was that there was indeed changes in cholinacetyltransferase in some areas, but the thing is that it wasn't associated to a loss of terminals that you could see histologically. So the fact that the enzyme is not expressed anymore does not mean that you've lost the terminals. It just means that there's some dysfunction, but it doesn't quantify any real cholinergic loss. So which target? So when we started looking at that, we had to decide about one target that we would think of. So first thing that was easy for us, we had no tracer for any of those molecules, so we weren't biased, if you want. We could start with a clean slate. None of the different targets that were presented in terms of receptors, acetylcholinetransferase, acetylcholinesterase, and cholinacetyltransferase were obvious candidates, but there was information that Katie will recognize, I guess, that came out of UBC, saying that looking at vesicular transporters could be a good idea. It wasn't for cholinergic studies, it was for dopaminergic studies, where the Pacific group showed quite a few years ago that the best way to quantify dopaminergic terminals in the basal ganglia was by looking not at fluorodopa studies, which were linked to synthesis of dopamine, nor to membrane cells transporters for dopamine with methylphenidate. The best way was to look at DTBZ, which is a ligand for the dopamine vesicular transporter, so kind of the equivalent of benzodiazepine is for cholinergic. And that conclusion was arrived at by the fact that the only one that showed a line of identity with the number of terminals measured histologically was actually benzodiazepine. So, you know, we got some kind of bias to our vesicular transporters. And also, we wanted to avoid SPECT. And the reasons for that are pretty clear. Lots of studies have been done with SPECT, but they're really hard to use. I mean, the kinetics of most of those tracers are extremely slow. So, for instance, in this study, where results were good, I mean, they had results that correlated with clinics and everything like that, but the kinetics were extremely slow. They had to wait up until 22 hours post-injection before being able to show results that look stable. So that's not a very good way of performing research studies. It's very demanding. People kept doing them. Results, again, with some molecules were somewhat better because we could probably reach some equilibrium after injection around, well, a pseudo-equilibrium, obviously, after some 6 hours. But again, 6 hours is long for most research protocol. It's difficult to convince patients to come with something like that. Still, people kept looking at it, came up with really interesting ways of quantifying the cholinergic system with iodobenzoazamicol, with SPECT. That was all nice, good results. But again, studies, even with new methods of quantifying this, using models that are basically those that we use in PET imaging, also graphic imaging of accumulation. Again, the problem was that you had to wait for hours before any kind of result could be obtained. So that kind of kept us away, especially given the fact that SPECT anyway is a much lower resolution kind of approach than it is with the PET. And although most people tend to consider SPECT as the cheap cousin of PET, in cases like that, tracers are much more expensive for SPECT studies because of the I-123 label, which is extremely expensive. So, you know, there are even parts of the central nervous system that never reach equilibrium with agents like that. So that's why we decided to go to this agent here, FeOBV, which was nothing new. I mean, 1993 was the first time it was described in terms of synthesis and characterized in tissues. And it was that indeed it looked like a pretty good tracer. And the same group came up with validation in animal models using autoradiography after IV injection, showing a distribution that indeed corresponded to what is known about the distribution of cholinergic terminals in the brain of rodents. And it worked pretty well. So we started working on it. The first thing we did is that we improved the way to label the molecule. The initial recipe, if you want, was really lousy. They got very low specific activities and very poor yields. So we worked on it and managed to get very high specific activities with pretty good yields. And That was initially in 2007 when we started working on it. And then exactly that, we got a scare. A paper came out, I think it's from France, where they studied a very similar molecule to FeOBV in rats and in monkeys. And what they showed was that there was very low penetration in the brain, of almost negligible. And there weren't huge variations across different areas of the brain, nothing close to what you should find for cholinergic systems. And moreover, there seemed to be a lot of defluorination. So basically, you got bone scans, not brain scans, not what we want. Okay, we got that scare, but then we used our own molecule and characterized it in rodents right after that paper, and we arrived at results that were much more encouraging. We had a distribution in rodents that showed that the highest values were in the caudate, which was expected, the amygdala, hippocampus, basal forebrain. Those are the areas where you expect to have that kind of accumulation. The study showed that there was no lipophilic metabolite, which is very important because that means that only the native molecule penetrates into the CNS, avoiding all kinds of complication when you want to model the distribution of the activity. And finally, the kinetics were very good. It goes in, stays where it should stay, and washes out from other areas very rapidly, which also helps perform those tests. This one is minuscule. I should have redone that one. We did 2 monkeys also because we wanted to see whether primates had a specific problem with defluorination, but we had great brain uptake in those 2 cynomolgus monkeys with no evidence of a significant defluorination. So we were reassured that we had something that seemed to be working. Actually, we were working on it and people at Michigan were working on it very hard. And it's always the same thing. You always want to be the first. But they managed this time to publish their monkey study before we did. But they had beautiful kinetics with uptake in specific areas that allowed us to hope that we could model the distribution again, measure the important factors like KD and things like that quite easily with this approach. And this was one of their monkey study with accumulation in the thalamus and the hippocampus. So, very good results. On our end of the deal, we started characterizing it in humans. I'll tell you one thing, we were not able to be the first to do human studies for one reason. We never got the kind of money we would have needed to do the initial toxicology studies to run by Health Canada and be allowed to proceed. So we worked with whatever we could have. We looked at rats in whom we had produced unilateral lesions, the nucleus of Minert, using this very nifty little thing here, 192 IgG safrarin, a very potent and very specific toxin for cholinergic neurons. So we injected it unilaterally in those rats, and we compared it to what happened in normal rats. And what was quite clear is that we had exactly the expected distribution. We already knew that, but also we had, I'll show you this here, excellent discrimination between the normal and the hypoactive side. I don't know if I can get an arrow here. No, not really. So anyway, the left side is on the left of the screen. If you look at the lower picture, the lesion was on the left side, and cortical innervation by cholinergic fibers is vastly decreased on the correct side. There's always a bit of a decrease on the other side because injecting the basal forebrain of rats, so it's very hard not to cross the midline somewhat, and those nuclei are just around the midline. But there was a very good lateralization of anomalies that we could show in 3D, corresponding really well to the expected areas of loss of cholinergic innervation. We tried to get close to humans, so we got to dead humans' brains. We have a nice bank at the Douglas Hospital in Montreal, where we have lots of normal and Alzheimer's type brains. And we did autoradiography in multiple regions and controls in AD, including the prefrontal cortex. And then we went to the hippocampus and to the striatum. The striatum, we were mostly looking at them to have binding in the internal capsule as a white matter area that could serve as a reference for computing of binding. We got interesting results. We had the expected decreases in CA1 and CA3 in prefrontal cortex, but then when we looked at the dentate gyrus, there was no difference. There was absolutely zero alteration of cholinergic terminals in the heart of the hippocampus. In fact, mild increase, no, not significant, obviously. So, what does this mean? Well, we got the expected deficits in the hippocampal region, but we didn't seem to have an effect on the dentate gyrus. So kind of surprised by that. We thought we were the first to show that. Never. You do a good literature search, and I'm opening a parenthesis here, and we'll talk about this. There was evidence a long time ago of hypersprouting, if you want, of cholinergic terminals in the dentate gyrus. And there was this study from 1985. And they did show with histochemistry that patients with Alzheimer's disease had really increased sprouting in the dentate gyrus. So we weren't the first to show that kind of plasticity of the cholinergic system in that given area. Why does it happen? Very good question. Is it a question of local, better trophic maintenance of cholinergic terminals? Is it because the subparts of the basal nuclei which project precisely to the dentate gyrus are somewhat different from other cholinergic cells in the basal forebrain? We don't know, but we found something ourselves that did correlate exactly with what they showed here, and therefore they concluded, as we had to, that there's evidence of some kind of plasticity of cholinergic systems in some areas of the brain. And remember that because I'll come back to something similar in an unexpected fashion somewhat down the road. How does this happen? Well, after that I've told you that beta amyloid is toxic to cholinergic neurons. I'm now telling you that actually it causes some sprouting of cholinergic neurons. That's kind of ambiguous as a statement, but indeed there are studies showing that Alzheimer's disease patients have sprouting of cholinergic in different brain regions, and you also get it in what they call and Lewy body variant of Alzheimer's disease, a term we no longer use. But there's a correlation between cholinacetyltransferase activity. So again, histochemistry, not very quantitative, but still, that's about as well as people could do back then, and the quantity of amyloid beta peptides. Well, it's not exactly something healthy, I would say. It's not that amyloid is helping those cholinergic neurons. The sprouting really is a case of sprouting of neuritic processes, processes that are really degenerating. So there seems to be some attempt by cholinergic neurons at getting back to normal numbers, but they don't really do it in a normal fashion. And therefore, there might be aberrant neurotic sprouting of cholinergic neurons, which is not necessarily functional. Another mechanism that could be involved in sprouting of cholinergic neurons is completely different. People have been looking, for instance, at another detour, at the catecholamine axons in the human brain in Alzheimer's disease, and repeatedly have shown that there's increases in terminals for noradrenergic axons in the hippocampus of patients with Alzheimer's disease. So that seemed a little mysterious initially, but it was found, by more than one author, and they indeed confirmed that there was sprouting, and what they identified was that the sprouting come from fibers, noradrenergic fibers that arose from the superior sympathetic ganglion. A little unexpected, but they did prove it and they did show at the same time that cholinergic hypersprouting in those areas is linked to that noradrenaline sprouting, because when they destroyed the upper cervical ganglions, they completely lost both the noradrenergic and the cholinergic sprouting. So indeed, there is a link between sprouting of cholinergic terminals and sprouting of norepinephrine terminals in the brain. Somewhat of a review here of those phenomena. So again, they state that there have been observations of sprouting of both systems. The noradrenergic one probably sprouts because there is initially a loss of cholinergic systems, which normally transport NGF back to their cell nuclei. So if you lose cholinergic terminals locally, there's probably an increased concentration of NGF in those areas, which allows the sprouting of the norepinephrine terminals, which in turn, by some mechanism which is not really clear at this time, will increase sprouting of acetylcholine neurons. So indeed, that's what we conclude as such. Moreover, this changes the processing of and the production of amyloid, APP, amyloid precursor protein, because the stimulation of target neurons in the hippocampus, at least by acetylcholine, results in activation both of muscarinic and nicotinic receptors on those cells. And those will have effects on gene expression, and in particular in expression of genes linked to beta secretase, which is one of the main enzymes which is involved in metabolizing APP halfway, if you want, because you need gamma secretase after that, towards all amyloid peptides. So just this, as such, will modify the production of amyloid beta in those areas. So that was all I wanted to say about the sprouting. But we have good evidence that it does happen, and we even have some ideas as to how it could happen. Amyloid stimulation, beta amyloid stimulation, norepinephrine stimulation, by mechanisms that remain to be explained. Okay, back to what we did with FUBV. Not only did we study imaging, we did also correlation between in vivo histology, then we don't have much choice. We did acetyltransferase immunohistochemistry, although again, it's not very quantitative, and imaging with FUBV in lesion rats. Same things, we used the same saccharin protocol to unilaterally destroy cholinergic neurons, and again you can see that there are controls up there, and then lesion rats with essentially unilateral losses of FUBV binding on the side of the lesion, so that's pretty clear. And histopathologically, we can show that there is indeed a large decrease on the left side of cortical ACH terminals as compared to what you see in the control subjects. And the correlation between what we measured in terms of uptake of FUBV with PET imaging and the density of acetylcholine terminals by histopathology is very high, confirming that what we're seeing is really exactly the losses of terminals that we produce with this agent, and whereas in normal tissues we see also the normal complement of this. We went a bit further. We started studying other cholinergic systems. We lesioned the that's a hard one to pronounce in English, the pedunculopontine tegmental nucleus, which is a brainstem nucleus composed essentially of cholinergic cells. And we basically just did what we had done with the other study at the nucleus of minor location, and we found essentially the same thing. So you can see that our lesioned animals have vastly decreased amounts of cholinergic cells in the pedunculopontine nucleus, and this results in a very asymmetrical decrease in the uptake in the terminal fields of those cholinergic neurons. So very high correlation again. So we do see exactly what happens from a histopathological perspective with that agent. Not only that, it does relate to the actual behavior of the animals. We used a behavior test that really measures the variance in the time it takes for the rat to make a selection between 5 different targets that he's presented with. And we measured the impact of lesioning of the pedunculopontine nucleus on the variance of that selection task, and compared it to PET imaging with FUVV again. So that's what we did, and again, very good lesioning, as you see on the right side of the microscopic slides. And as you can see, as the number of terminals decreases in those animals, there's a vast increase in the variance of their response time to the selection test, showing that there's a component of dysfunction that is explained by histopathology. And at the same time, we do find those lesions in the pedunculopontine nucleus on the side that has the lesion. And oops, I thought I had the slide with the correlate. Well, anyway, the correlate, yeah, that's the slide. We have a very strong negative correlation between the variance, which is increased in normal animals, and the number of cells that are left as measured with FUVV. So we measure histology, and we measure the behavioral equivalent of histological loss of neurons and cholinergic systems with that approach. Okay, so everybody wants to do mice for a while, but after that, it's time to go to humans. And as I said, we weren't the first to do it. The group in Michigan was the first. In 2014, they came up with 10 patients that they injected with FUVV, which, surprisingly, that's the way Health Canada works. As soon as a paper had been published on humans, even if it's a low number of subjects, as long as nobody died, and nobody died, which is a good thing, then we were allowed to start using FUVV for research purposes. So that was a green light for us to start doing patient studies. And in fact, the one thing we got from that paper is that we set a limit to the mass of FUVV that we would inject at most in subjects based on the maximum dose they give to any subject, and this didn't result in any sign of toxicity. So we used that after they had published studies. Great kinetics, great distribution, exactly what we hoped that they would, well, that we would find, but that they did find. Okay, so we got back to work and said, okay, we won't have the first normal subjects, but we started a study in Alzheimer's patients using FUVV, a small study. I mean, it's hard to do those studies, 12 participants, but the results were absolutely spectacular. Those three columns represents imaging with FUVV, second one FDG, last one with NAV4694, which is a ligand for amyloid. And the intensity of the deficits, and we have the same scale in terms of standard deviations from normals. As you can see, the amplitude of deficit is way larger with FUVV than it is with FDG or with amyloid imaging. So it seems that we could have something that could be extremely sensitive, probably even to very early anomalies in patients with Alzheimer's disease, where I'll tell you about what we're doing furthermore in a moment. But this was very encouraging. Actually, it was named one of the 10 discoveries in Quebec in 2017. So we at least got this little compensation from not having been the first to do normal subjects. So what we're doing right now is that we're increasing our cohort of 80 subjects. We need more. I mean, we don't have that many. We're up to about 15 right now. We will evaluate it in terms of its correlation to the clinical presentation of the subject's cognitive performance. And we will try to follow the time course of acetylcholine losses by doing patients of different ages, from very young patients all the way to patients in their 70s and 80s. And we hope to detect the normal variations that might happen. Even with the limited data that we have, we do think that we have a tool that's going to be very useful for assessing extremely early cases of Alzheimer's disease, and which will allow us to better confirm the depth of involvement of cholinergic systems in neurodegenerative diseases. And we've also moved on to something a little different, to Parkinson's disease, and actually not really Parkinson's disease. You'll see what I mean by that. There was this review paper which reminds us that when Lewy described his Lewy bodies in patients with Parkinson's disease, The one thing that he noticed was that the area with really the most intense accumulation were the basal forebrain nuclei. So cholinergic neurons again seem to have this weird sensitivity to abnormal proteins and accumulate them maybe more than other neurons. There had been results using other types of traces before showing that indeed you could show anomalies of acetylcholinesterase levels with PET. So yeah, there were indications that it was involved. There were studies showing that people who have cognitive disorders accompanying Parkinson's disease, and you have a large number of subjects here, did indeed have abnormal binding both for dopamine in the striatum and for acetylcholinesterase in other areas of the brain. But the interesting thing was that the anomalies with the cholinergic systems were increasing very monotonously as compared to the symptoms of the patient, whereas the dopaminergic anomalies seem to appear and then remain more or less stable. So again, that tends to suggest that what happens in terms of cognitive deterioration in Parkinson's disease is in large part related to cholinergic anomalies. Again, Michigan, very nice, although small number of studies, just like ourselves, so we're not going to criticize them for that. They did studies in patients with dementia with Lewy body and found significant decreases of FeOV binding in the basal ganglia, in the brainstem, and some cortical areas also. So again, exactly what was expected given what Lewy had said about the accumulation of Lewy bodies in cholinergic cells. What we decided to target was something before Parkinson's disease. We know that there are different conditions, two essentially, that kind of are predictors of incipient Parkinson's disease. One is what's called REM sleep behavior disorder, which is a condition where people, when they start having dreams, REM sleep, instead of having atonia of their muscles, keep their muscle tone and start enacting in their dreams, giving sometimes dramatic results of broken noses and partners and things like that. So we know that it's linked to incipient Parkinson's disease. There's also constipation, something that even Parkinson's had described in this patient, sometimes dating back to 25 years before they became symptomatic. The advantage that we had here is that we had the largest cohort in the world of REM sleep behavior disorder in Montreal. Right? And this guy, Jacques Montplaisir, has really published the textbooks on that. All the criteria are the ones that he published. So we had access to that cohort, which shows that people with REM sleep behavior disorder over a period of 10 years convert to at least 80% to Lewy body pathology, including Parkinson's or potentially dementia with Lewy body. So, and as you can see, it's a pretty steep increase in the number of patients with that kind of disease. So we decided to look at those so-called idiopathic REM sleep behavior disorders. That means those that don't have an obvious Parkinson's disease or Lewy body dementia. And we found pretty interesting things. Again, it's a small study. We just got it accepted in sleep. 5 controls, 5 REM sleep behavior disorders. And this time, what we found was there was significantly increased FeOV binding in multiple areas. You can see it here in the brainstem, nuclei, deep nuclei, the cerebellum, and some cortical areas. That's really what we observed. So, brainstem, we had the reticular formation of the medulla, the region of the ceruleus nucleus, and then the tegmental periaqueductal gray matter, and then we found also anomalies in the thalamus, deep cerebellar nuclei, some cortical region. Interesting. Again, early on in some kind of process, there seems to be some kind of pathology that produces increased innervation. But in this type of innervation, again, it does not seem to accomplish what you would expect cholinergic terminals to accomplish, because the more increase we had, the more symptoms the patients had. So there is this tendency to cholinergic systems, apparently, to start sprouting in all kinds of weird ways that do not bring about compensation, but that seemed to just characterize a reaction of cholinergic systems to incipient pathology. Constipation? Well, that's a hard one to study. We've done a series of patients. We know that indeed it does happen for a reason. There are Lewy bodies that can be found in parasympathetic sacral neurons, so that's not a big surprise that it would affect patients in that way. As I said, Parkinson noted that. It might even be, I mean, I have to mention it, everybody mentions the microbiome, and it seems to be related indeed, at least in animals, to the presence of a certain kind of bacteria mix in the colon, which might probably retrogradely reach the central nervous system by the fibers that innervate those areas. So we've started doing patients that we study with FEODV for Parkinson's and for Remsley behavior disorder. And we've been desperately trying to find a way to quantify uptake in the guts. But, I mean, it's very hard to do that. I mean, drawing regions of interest that make any sense is complicated. The protocols that we have call for early imaging of the central nervous system. This is an add-on. We know that there is intense, you can see it, liver excretion of the tracer. So at some point, what we'll be seeing in the guts is not gut uptake, it's going to be bile, most likely, that we'll find. So far, we haven't really managed to quantify this, but we're trying hard, and maybe we'll arrive at something at some point. Okay, just a few words about what we could do with you eventually. We have this pretty nice FET-enabled pharmaceutical. Those are well established. The half-life is not so bad, 110 minutes. So you can actually ship agents labeled with F18, and we've done it repeatedly to Quebec City, which is even more of a drive than Montreal to Ottawa. The thing is that we've done it for mostly amyloid agents. FEOBV is something a little different. We don't have a real therapeutic index. We know that it's highly toxic as such, as a benzoic. We've had the initial studies that we did on animals when we had a big batch produced in the morning. The animals that got injected later in the afternoon died. So obviously it can happen if you have low specific activity. So we'll have to work it out and make sure, do the calculations and look at how long it takes for us to ship it from the neuro to here, for instance, if we want to start doing studies with it. There might be an alternative. People have published a few years ago a series of PET tracers that are labeled this time differently. Instead of using F18, they use brome as a radioactive form of brome, a PET-emitting form of brome. And in particular, there's one of the derivatives that they had that could be labeled quite easily. One-step labeling, pretty simple to do. Brome has a 16-hour half-life, so that would make it easy, actually, to ship the molecule over relatively long distances. It's even better than I-123, which has a 13-hour half-life, and you can ship I-123 from Vancouver to Montreal and things like that. So this one could be of interest. There's been a paper talking about easy, quote-unquote, production of brome-76. It's not that easy. The reaction is that we bombard selenium-76 with protons and we get brome-76. But the problem is that it uses a so-called solid target. What we do is in chemistry most of the time, well, at this point only chemistry we do at the neuro, is on liquid targets and some gas targets. Doing solid targets is a bit more complex. We don't have much experience, but we could think about it. So if there was an interest in building a larger program, that's one of the things that we could think about that would allow us to ship long distance traces of this nature without worrying about having to inject too large a mass to get the minimum activity that we need, which could result in some toxicity. So that would be one way of eliminating that. Okay, that's what I wanted to talk to you about. If you have any questions, I'll be happy to take questions. I'm on time. That's good.
Thank you. There's so much interesting work there and so many interesting directions to go in. But I'm thinking of the REM sleep behavior disorder, this paradoxical finding of increased signal. Do you think this has a parallel with what people often find with BOLD and fMRI, that there's this paradoxical increase in BOLD as people go from normal to MCI, and then it drops off when people
later
go clearly into the dementia?
That's a very interesting question. With I do 2 days a day of clinics. I do FDG PET studies and neurodegenerative conditions. Everybody agrees that the standard type of Alzheimer's disease, the amnestic one, is related to anomalies in the default mode network. And the medial temporal region, the hippocampus, is very much part of that system. In FDG, we have exactly the same thing. We don't see a decrease in FDG, except for very early cases where we might have a slight decrease. So it seems that, again, there's some kind of a, kick-in mechanism that boosts the activity, which is reflected with blood flow studies, actually, with MR. So yes, I think if you have acetylcholine in a region that's liberated in a somewhat uncontrolled way, it is known that liberation of ACH in the cortex will result in increased blood flow because there's an increase in amino acid, excitatory amino acid transmission. So yes, I think it might be part of the explanation, this increase in cholinergic terminals, although again, some reports say that they're not normal terminals. So we don't really know if those increased terminals really release more acetylcholine, but it's a possibility.
So there's one way to try to answer these questions to do longitudinal studies, which then double or triple or whatever your...
Yeah, we could do MR, do BOLD studies and acetylcholine terminals imaging. We could, now that you mention it, we have a protocol that we've been using for years for dopamine studies with raclopride, where we stimulate dopamine release with whatever protocol you can think about, and we can show that the amount of raclopride that binds to D2 receptor goes down because dopamine is released, and therefore you have a competition that's higher. So maybe by using one of the well-characterized receptors for acetylcholine ligands, we could replicate that kind of thing and see whether indeed whether there is indeed increased acetylcholine in those regions. That would be a very interesting thing to do. Yes?
About the finding that the FAG and the FEOBV correspond regionally, did you look at correlation?
I did not say that exactly. I said that in the mesial temporal regions, we usually don't have a deficit with FDG, which is kind of surprising. I mean, the region shrinks rapidly, but we know that there's increased blood flow. And we also found that the dentate gyrus indeed shows more FEOBV binding. That's the only place, and we don't have correlations between the two. Those were not done in the same. I mean, there might be a correlation, but we haven't tested it. There might very well be.
Different people.
Yeah, they were different people. Yeah. Okay. We haven't done, well, we've done FDG studies in Alzheimer's. Actually, we should go back and look at those 6 Alzheimer's and 6 controls that we did with FEOBV and FDG and NAV3. We didn't really try to do correlation between FDG and FEOBV. Good idea. We'll do that. And I'll let you know if it works. And if it does, we'll include you in the paper. So. Yeah, that's a good idea. Yeah. Yes?
Is it true that you can't measure binding in the cardiac and the striatum with FEOBV?
Oh yeah, we can. There is a significant, the striatum is an area of high binding, which is...
Does it equilibrium in time? Or?
No, it doesn't, actually. There's probably too many cholinergic neurons in there to actually reach equilibrium. But yeah, I mean, and of course it's a different case in the sense that there's tons of local cholinergic neurons instead of long projections, which do exist too, but a lot of is local interneurons, no, cholinergic neurons.
Yeah, no, I just bring it up because the prior study that may happen here with schizophrenia, that's a region of great.
Yeah, I mean, yeah, Laurie and I have been talking about that. I really have no expertise in the field of schizophrenia, but I mean, it seems from what all the studies we did in animals, it really seems to be following cholinergic neurons very tightly, very well. So yeah, I mean, I think it's certainly worth having a look.
So even we could look there if we scan mine? Or?
Sorry?
So could we look in the striatum?
Yeah, absolutely. There's it's taken up there. So far, the pathologies that we've looked at, we didn't find. Well, we looked there and we didn't find anything different, really. But yeah, in a disease that, is, from what I hear, is expected to affect cholinergic innervation in the basal ganglia, I think that FUBV has a very good chance of being able to pick that up.
Did you look at your uptake curves?
Sorry?
I'm just wondering how far you looked at the uptake curves when you're saying that
Oh,
it never reaches equilibrium, but what was the latest time you scanned?
Good question. I don't remember.
Okay.
But the Michigan study probably has that. The first one by Petru in 2014.
Because then I thought some of their images, it was just white and
Yeah, I mean, there's extremely intense uptake. You know, the same thing happens with raclopride when you look at, well, no, the inverse, sorry.
Yeah.
The same thing happens with falipride, which you can use for looking at the cortex, but not at the basal ganglia, whereas raclopride is good for the basal ganglia because it has lower affinity. So, but we haven't really tested the basal ganglia. Yeah. But it's, yeah, it would be another nice potential application.
Did you look at the heart?
We have the heart, yes, we did. Actually, when we do the abdominal imaging, we always want to make sure that the inferior portion, the inferior wall of the heart is included in the field of view. So,
Not the first few minutes, like you don't have the.
no, but that's it. You see the heart is present
Yes.
here, 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 here. We don't see it that well, but it's still there. But that's the big problem. Again, we do the brain initially and we follow it over the first hour. So the first images that we really do in 56 minutes here are approximately 1 hour post-IV. I mean,
So do you think it's not?
that's part of the problem we have with quantifying those organs. I mean, at some point we could start a protocol where we would target specifically the heart and the abdomen, but it would have to be funded separately because every investigator is looking at the brain mostly, not the peripheral.
What do you do with the brain?
Well, those were done in the patients with the REM sleep behavior disorder. So the goal of the study,
Why do you do the brain first?
because we want to have the kinetics in the brain, because we haven't done that many subjects, so we're trying to acquire the first hour of kinetics in the brain to make sure that it's that we have everything under control with those patients. But yeah, at some point we'll probably stop doing a full hour. We'll probably do just one scan at a later time point, and then we could start doing something more serious with the abdomen. But so far we haven't gotten there.
So what we thought about here is doing a late scan and the first.
Yeah, and I think that's going to work because the kinetics seem to be just fine for something like that.
Have you compared SUV values, like SUVRs versus your BP values in the
No,
brain?
no, I don't think so. I don't remember having done that.
That would be right, that's kind of, we would be doing SUV.
You would be doing SUVR?
Basically, if you want to show that that grows well.
Yeah.
Well, yeah, I don't know.
We've done full kinetic modeling on our cases right now, so we get binding potentials.
Okay.
Yeah. Yes?
Could you just give some advice or, comment and give advice on what seems like a possible problem for all of us, where if you want to find a basic effect, say see a difference between people with a disorder and not, an N of 5 or an N of 10 is sufficient.
Well, we've found that for Alzheimer's.
Yeah, and the effects are clear.
Yeah.
But then when you want to start to relate signal to behavior, And especially combine predictor variables, then all of a sudden you might realize, oh, we need 200
Well,
participants or
Yeah.
what have you.
in the lesion rats, we did it on 15 or 20.
Yeah.
But those had, I mean, they had massive losses. I
Right,
mean,
and, so,
that's,
and you saw some clear relations with
Yeah.
behavior, but how do you navigate that? You know, you need a lot of money, you need to take a lot of time, but you want to be ambitious. How do you solve this problem? This trade-off?
oodles of money, basically. I
Alright,
can't
so
say.
it's more money, always
Yeah,
the...
I mean, you know, those molecules cost a lot to prepare.
yeah.
I mean, the synthesis, we have it down pat, but they're long. The precursor is expensive, and so, yeah, that ends up costing, I mean, yeah, that's one of the banes of pets. That's expensive.
No, it's cheaper.
Yes, which is why, yes, it's...
Yeah, we should. I mean, even for just increasing the number of normals that we study. I mean, Laurie has been looking at doing normal subjects. I mean, if he does a few of them, that increases our bank of normal subjects by a significant proportion. So we'd be happy to you know, eventually share results in normals and things like that, because we have limited data, older people only, we don't have very young subjects, and we need to do that. So yeah, we're really looking to, yeah, multiple groups can each get some of the money necessary.